Hello students, welcome back to a new video of Sunanda's tutorial. So today in this video we will discuss about the photorespiration. It is a very important topic of your plant physiology, and this topic is present in your plus 2 first year, plus 3 syllabus as well as MSc botany syllabus. It is a very important topic for NEET aspirants as well as SSB, TGT, PGT, even for OPSC assistant professor aspirants. So without wasting any time, let's start. So at first, earlier in the year 1920, Otto Warburg observed that the presence of excess oxygen in the atmosphere inhibits the photosynthesis process in green algae. So whenever there is an excess amount of oxygen present in the atmosphere, it will adversely affect the process of photosynthesis in green algae. And later on, this inhibition was also observed in several green plants. Thus, inhibition of photosynthesis by high level of oxygen was termed as Warburg effect, according to that scientist's name. However, this inhibition was observed mostly in C3 plants. So, the plants which perform the C3 cycle and where the first product or stable product of this carbon dioxide fixation is a 3 carbon compound and phosphoglyceric acid, and that plant is known as C3 plant. So mostly the C3 plant shows this type of inhibition. So C4 plants were hardly affected by varying O2 concentration. Extensive studies were then carried out to explain this Warburg effect, and these studies led to the discovery of the process photorespiration. The process of photorespiration was first demonstrated in tobacco plant by Decker and Theo in the year 1959. Since then, it has been reported in a number of plants, particularly those which perform the C3 cycle. The process is defined by Krotkov in 1963, and he defined this process as an extra input of oxygen and extra release of carbon dioxide by green plants in presence of light. So he observed when there is an extra input of oxygen or extra uptake of oxygen by the plant occur, there is an extra release of carbon dioxide by the green plants. Before discussing the actual biochemical mechanism of photorespiration, let us discuss the first step of C3 in some details. So this is the reaction where ribulose 1,5-bisphosphate combines with carbon dioxide to form 2 molecules of phosphoglyceric acid in presence of the enzyme rubisco or ribulose 1,5-bisphosphate carboxylase oxygenase. And this enzyme rubisco has the remarkable property of fixing not only CO2 but also oxygen. Hence the name of the same enzyme performing dual role is RUBP carboxylase as well as oxygenase, and rubisco in short. However, this enzyme has much greater affinity for carbon dioxide than O2, but it can combine or it can fix both carbon dioxide as well as oxygen to the plant. Then the process of photorespiration, as rubisco is a dual enzyme or dual natured enzyme which acts as carboxylase in high carbon dioxide concentration. It can act as oxygenase in high oxygen concentration. Rubisco is the most abundant protein or enzyme in the biosphere. It constitutes 16% of chloroplast protein. So about all proteins, from all the proteins which are present in that chloroplast, 16% of all the protein which are present inside chloroplast is the rubisco. Photorespiration is a cyclic process which requires 3 organelles to complete. For example, first one is chloroplast, second peroxisome, and third mitochondria. At first, chloroplast in high oxygen and low carbon dioxide concentration, rubisco acts as oxygenase and oxidize the RUBP into 3-phosphoglyceric acid, that is PGA, and phosphoglycolate. Phosphoglyceric acid have 3 carbon compound, have 3 carbon in them, and phosphoglycolate have 2 carbon in them. phosphoglycolate is dephosphorylated to form glycolate or glycolic acid. The glycolate is the substrate for photorespiration. Hence, this cycle is otherwise known as glycolate cycle. Okay. Next, in peroxism, so after the first fixation in chloroplast, the RUBP oxidized to form phosphoglyceric acid and phosphoglycolate. Then phosphoglycolate, from phosphoglycolate, glycolate is formed. Then in peroxism, glycolate react with oxygen to form glyoxylate. Then glyoxylate is aminated. That means one amine group is added to glyoxylate to form glycine. So in peroxism, glyoxylate and glycine is formed. Then in mitochondria, two molecules of glycine combine to form one molecule of serine with release of carbon dioxide and NH3. Then serine again enter into peroxism where it is deaminated to form glycerate, and finally glycerate moves into the chloroplast and enter into Calvin cycle. So it is a cyclic process and require three organelles. One is chloroplast, second peroxism, and third mitochondria to complete. Okay. So C4 plants generally do not perform this photorespiration because rubisco is confined to bundle cells in C4 plants, and C4 cycle operative only in mesophyll cell pumps CO2 in the form of C4 acid, so that CO2 concentration remain high in the bundle cell. This ensures the rubisco functions as carboxylase, minimizing the oxygenase activity. So this process is a highly wasteful process which do not produce any energy, rather it consume many energy and finally producing the carbon dioxide, which is very harmful for the atmosphere. So it is the whole process of photorespiration. I hope you really enjoyed this session. In my next video, I will discuss another important topic of plant physiology. So please stay tuned, like my videos, share with your other friends and subscribe to my channel Sunanda's Tutorial. Thank you.